Hi, everyone. Welcome to today's EMSL LEARN webinar, where we are going to be discussing EMSL's new 10-year initiative, the development of a molecular observation network for soils, water, and biogenic emissions to air. So was curious to see, where is everyone coming from? In the chat, let us know where you live. That would be great. So I'm here in Richland, Washington, and I know from previous webinars we've got folks from all over the world. So we love hearing from where you're joining us from. So next slide, Roxy. So on October 26th, the next EMSL LEARN webinar is going to focus on the nuclear magnetic resonance analysis of complex mixtures, 3D structures of big and small molecules, and the characterization of multiple phases. We will be sharing the link for you in the to register in the chat later on in today's webinar. But go ahead and keep in touch with EMSL by subscribing to our email and social media channels if you haven't already. The links will also be dropped into the chat. This is a great way to keep up to date on all of our open call for proposals and other opportunities to work with us. Roxy, next slide. So today, EMSL Earth scientist Emily Graham is going to give an overview of the purpose, the vision, and the goals for the MONet initiative. And EMSL Earth scientist and postdoctoral researcher Maggie Bowman will be walking you through the different data types. She'll be walking you through the different data types being analyzed and collected through MONet, and is going to discuss a case study with analyzed data from MONet's recent 1,000 Soils campaign research pilot. And then as well, John Bargar, EMSL's Earth scientist and the lead coordinator for the MONet initiative, will be discussing opportunities and timelines for you and your organization to participate in MONet. There will be time at the end of today's webinar for questions, so we encourage you to post your questions in the Zoom chat, and Emily, Maggie, and John at the end of their presentations will answer them during the Q&A. And so with that, we will begin with Emily. Take it away, Emily. Thanks.
Awesome. Hopefully these gray squares will go away here in a second. My name is Emily Graham. I'm the Integrated Research Platform Lead for Biogeochemical Transformations here at EMSL. We're really excited you guys are here today. So I'm going to kick us off with a little bit of an overview of our molecular observation network, which we're calling MONet. As Linda said, this is a 10-year initiative. We just finished a pilot, the 1,000 Soils pilot. That was a one-year effort that Maggie will talk about in more detail to help us get this up and rolling. And we're really excited to expand some of the things we're doing and ramp us up over the next 9 years or so. So we're really motivated by a central challenge that climate change occurs and is predicted at regional or larger scales, but it's really underlain by molecular processes that dictate biogeochemistry at molecular and local scales. And so it's really important to focus on these mechanisms, not only at individual site locations, but also in a very standardized way across regional or larger scales that we use to predict changes in our environment and changes in future climate scenarios. And so with this background in mind, we are launching MONet. Oops. And I lost control of the slides here. Oh, there we go. And goodness. 1 second. There we go. So we're launching MONet in the vein of other ecological networks that have had tremendous success. So you can think about networks like NEON and Ameriflex that are doing a tremendous job of making data open and available, as well as newer efforts such as our Wonders network here at PNL that really focus on river corridor systems. And those of you who know me know that I'm a huge fan of open networks because I really think they can shift the way that we as scientists start to think about addressing our scientific problems. So if you think about the way you might address a problem as an individual researcher, you might start out with big visions and big dreams and huge hypotheses. And at some point, you're really going to get constrained by the budget or the resources that you have access to. But by crowdsourcing science and having these open networks, we can really change the way we start to think about science. So from a perspective that's really like, what's the best I can do to address this question, given the resources that I have access to what is the best way that we can address our scientific objectives. So with that in mind, the vision for MONet is really to enable, sorry, there's a delay in the slides here, is really to enable transformational understanding of the drivers and responses to environmental change. Not only us as scientists at EMSL, but also throughout the broader DOE and even citizen science and other funding agencies as well. So far, we've focused on 3 aspects of this through our 1,000 Soils pilot. We're working on generating high throughput standardized workflows for molecular characterization of environmental samples. So far, this is really focused on organic matter characterization as well as physical characterization of soil cores through X-ray computed tomography. And I think we made a lot of great progress here in the last year and are continuing to make progress. Then all this data is going into an open database with molecular data, probably by the end of this year, so about a year from now. We'll have that up and running, and we'll have intermediate data types. Data from our first year should be available here towards the end of this calendar year, beginning of next calendar year. And our goal with this is to enhance predictive models as well as fundamental science. So we need to better understand the mechanisms of biogeochemistry and then be able to integrate it into some sort of predictive framework. And we have some really nice multi-scale modeling frameworks available here at EMSL as well as scientists to support that integration. So looking at the broader vision, we have 5 sort of key areas of MONet. The first 2 we've done a lot of work on in the first year here of MONet. Thinking about automated organic matter and soils analysis, traditionally a limitation here has just been that it takes a lot of time to generate molecular data. So we've done a lot of work on standardized workflows, increasing throughput, and actually we're about halfway through a large initiative to automate a good portion of these analyses. We've also been generating a network of field sites. So in our first year, we worked heavily with NEON as well as individual collaborators to generate a network of field sites that we can go back to. We're definitely interested in expanding this collaboration as well. And then things that we're also working on that maybe are a year or 2 out are deploying a network of field sensors, and not only standard sensors that may come to mind, but also those that can detect changes in molecular processes. So some of these newer optical sensors, as well as IR sensors for measuring carbon chemistry, things like that. In-house, we're developing a lot of rhizosphere sensors. That's led by Amir Akami, who's the IRP lead of our rhizosphere function initiative. Again, targeting biomolecular molecules or metabolites there, as well as specific microorganisms. And then finally, an ongoing effort is our model experiment integration. And that's led by my colleague, Satish Kara, who's the IRP lead for that research area. And the slide should change here. Okay, so I referenced this several times, but our 1,000 Soils project was really focused on the soils component of MONet, which is our near-term goals, just because we can't do everything all at once. We do plan to expand into water and aerosol systems, but we're starting with one thing and trying to do it well and then ramp up. So this has really motivated our focus on soils, on the tremendous amount of carbon that these below ground systems harvest, as well as our poor understanding of how carbon cycles in these systems, and the encouraging work that other molecular studies have done on soil organic matter. We also know that climate change has huge impacts on soils, changes everything from the types of chemicals that are deposited into these environments, to changes in soil microbiomes, changes in soil physical structure and hydrology as well. So I'm just going to show an overarching workflow, if my slide will advance, from our 1,000 Soils pilot. Maggie Bowman will go into this in more detail, so you do not need to understand everything that's on this slide. But basically the gist is we developed standardized sampling kits, included core materials, standardized protocols, everything you would need to collect a soil core. We sent that out to collaborators all across the country. They took 2 cores for us and shipped it back to EMSL. We did all analyses here. One of those cores went through a pipeline for biological analysis. So things like chemistry, microbial function, so enzymes and metagenomes there, as well as organic matter profiling. The other went through a pipeline to do hydraulic properties and structural characterization through FCT. So Maggie will give you more details on that workflow. I also wanted to note that here in the next week or so, we will send out a flyer with some more information on the specific data types that we're collecting, as well as we'll have a presence at AGU. So we'll definitely send out when our talks are, hoping to have a mixer at AGU as well, if you want to get together and chat about any sort of details on that. So with that, I will hand it off to Maggie.
Perfect. All right. Thank you, Emily. And so I'll just proceed forward with some of the MONet methods, as well as a case study from our early 1,000 Soils pilot research. So
Maggie, are you able to turn your video on?
I can figure that out, but not while I have control of Roxana's screen.
Okay, go ahead.
No, I'm so sorry. There we go. Awesome. So I really wanted to highlight that soils are heterogeneous and diverse. And one of the big things I want to focus on here is that you can see that there's different like yellows and greens and blues. Those are pool noodles, which is kind of a unique solution to prevent the cores from sliding around while shipping. And one of the things that I want to really focus on and draw your attention to is that there's some cores here where we didn't get the complete core, or we have incomplete depths. And so this 30-centimeter core that we're trying to collect in some areas isn't able to be collected. Emily commented a little bit on the traditional ecosystem networks. And she commented on both Neon and Wonders, which are some of the more, and Meriflex, which are some of the more advancing networks that are more open data types. But oftentimes traditional ecosystem networks, such as the long-term ecological research sites, or the critical zone observatory, are led by individual PIs and research institutions, are often limited with standardization between the different networks. And finally, they focus a lot on the macro scale measurements. And these are some examples of these would be texture, carbon, and respiration. One of the steps that we were trying to focus on for the 1000 soils pilot for MONet was, how do we increase these standardized measurements, but also how do we include more field sites than are able to by these limited networks. And so one of the key motivations for MONet was that the current ecosystem networks lack these molecular level measurements to advance Earth system models. So I'm going to put up a map here where we're showing the different types of collaborations that we've worked with, where blue is academic, green are the critical zone network urban sites out of the East Coast. We have some summer school participants, which are yellow, as well as all of the purple sites are neon, and then we have partnerships with the science focus area sites in orange. And so we have quite a breadth of sample sites included in this site, ranging from Hawaii, which isn't on this map, or Florida to Maine. So when we were setting up MONet as an observation network, we kind of had to figure out what data was most important. So we needed to identify what measurements are needed. And so for these, we decided to focus on molecular level measurements, such as metagenomics and high resolution organic matter composition, as well as micro and macro level measurements. The big one that we're adding here is going to be the XCT, or X-ray commuted tomography, as well as including metadata across every step of this process. So that's the field sample prep, instrument data, and data analysis. And so finally, our last question was, what capabilities are unique to EMSL, and what do we need to bring for soil analyses? So I'm going to put up this slide, and it's going to be a bit overwhelming at first, but we're going to walk through it slowly. And so I wanted to present all of the measurements that the 1000 soils pilot completed. Starting with, at first, we have biological measurements. So that includes metagenomics, respiration, microbial biomass, as well as elemental species for nitrogen and phosphorus, and enzymatic activity. Next, we have what I would broadly classify as chemical, which is going to be the macro and micro nutrients, cations, total carbon, and then organic matter composition. And finally, we have the physical, or kind of point-based measurements, which are going to be the field measurements, which includes things like infiltration and temperature, bulk density, as well as pore size distribution, volume, and connectivity, texture, pH, gravimetric water content, as well as we have water retention curves over 3 saturation ranges, including saturated through wilting point, as well as the water potential. I really also want to emphasize that each one of these measurements we have to work to develop a standardized method, output, and metadata collection. So this is another way to visualize this. And these are the measurements that are unique to MONet compared to other networks. And so we are going to be providing metagenomic sequencing as well as organic matter composition using combined FTI-CRMS and LCMS, as well as using tomography to get the pore size distribution. And then we're adding in the hydraulic properties through the water retention and saturation curves. So as Emily mentioned, part of the 1000 soils pilot for MONet was to develop a field sampling protocol as well as a field kit. Here's a little bit of an image of the kit. What it looks like is we have a large Pelican case that has all of the standardized coring equipment needed to collect a standardized sample that is a 10 centimeter by 30 centimeter soil core. We also include, knowing that sampling doesn't go as we planned, we have backup plans included as part of our kit. So if we aren't able to collect intact cores, we collect augured cores, or at worst depth stratified cores using shovels. So the base core processing workflow, and this is a little bit simplified, starts with cores are received and assigned unique identifiers. In the field, we collect one large biotic core, which will be represented by a purple pipeline, as well as one abiotic core. For this demonstration, that's going to be an orangey yellow, and 4 smaller cores. These smaller cores are used to capture the heterogeneity of the surface soil around the field site that we're at. With this, the biotic core is depth segmented into the top 10 centimeters, bottom 10 centimeters, and middle 10 centimeters. And then next, we homogenize the top section with the three of the 4 smaller cores, and we collect fresh samples from the top and bottom sections for DNA, microbial biomass, nitrogen and phosphorus extracts, as well as pH and gravimetric water content. And then finally, the remaining soil is air dried for other analyses. Moving back over to that abiotic core, we do low resolution XCT scans collected on the whole core. That's collected in 2 image sections, and then those are stitched together, as well as we do a high resolution scans on the top and bottom sections of the core to collect more detailed information. And then finally, that core is depth segmented, sieved, and air dried before undergoing hydraulic property measurements on the top and bottom sections. So the metagenomics are sequenced by JGI or GeneWiz, and they're done on fresh soil stored at -80 degrees Celsius. And we're collecting 45 gigabytes per sample using the Illumina NovaSeq technology. We are collecting high resolution organic matter chemistry through Fourier transform ion cyclotron resonance mass spectrometry. That's unique because it gives us thousands of peaks, but it tends to miss some of the smaller, low molecular weight compounds, in which we are doing a liquid chromatography mass spectrometry method to capture those missed compounds from FTI-CRMS. All of that data is being processed using EMSL's newer software of Core MS. The XCT measurements, which give us the pore network structure, soil moisture, range from 50 to 200 micron resolution from the high range to low range. And so when we get the low resolution scans, we can slice a scan of the core, and we can visualize the core where the intensity of the darkness is related to a density. And then we can as well do high resolution scans where we are looking at the pores, connected pores, and unconnected pores collectively, as well as we can do that for the bottom sections of the core. So this is providing us with a lot of information about soil pore networks, as well as can be used in hydraulic modeling. We are collecting hydraulic properties using an off-the-shelf set of instruments to measure the saturated conductivity, or the KSAT, as well as the water retention and unsaturated hydraulic conductivity that's collected using the HYPROP2 from the meter group. And then finally, the third is the water retention and wilting point using the WP4C dew point potentiometer. This data is then used by Mark Ruckhold to inform hydraulic modeling, and then we can kind of get some of the bigger picture or site-specific hydraulic measurements from there. So I really wanted to focus, and I stole this slide from John, and you might see it a little bit later, But the MONet project works as part of the EMSL user project. And our goal for this really was to develop the standardized sampling across the US. And as Emily mentioned, we're moving into the rise of air sensors and soil automation. But all of this, kind of continues to feed back into itself. And so I'm going to focus next on a use case that we're working on as part of the 1000 Soils pilot. Here we're working with the Confederate Tribes of Warm Springs, as well as researchers that are based at Washington State University. We are working with them over 3 field sites on the tribal lands, one which experienced multiple high severity burns in 2003, 2014, and 2020, with moderate severity burns in 2014 and a high severity burn in 2020, as well as one that has had no recent burn history. Another way to think about, and here we have a cute little video of our sampling kit that unpacks and repacks itself. That was put together by our comms team. So we sent them the kit and they did some fantastic field sampling and then brought us back the soil cores. And when we received the soil cores back in the lab, we processed the biotic cores. And I wanted to, I will continue with this gradient where we have the high severity burn being presented by 3 fire symbols and the low or no severity burn being represented by a fire with an X through it. And these are images of the biotic cores that were processed immediately on arrival back at EMSL. And what you can see is visually that there's large differences in the soil structure and texture that we can get just by having these images alone. But in addition, there are unique chemistries. So again, I'm going to walk through the biological pipeline super quickly. So we depth segmented them and we measured a bunch of different measurements, as well as we outsourced measurements to Oregon State University and KUO Testing Lab for respiration, enzyme activity, and soil chemistry. And the metagenomics for these samples were sent out to GeneWiz. And what we found was that there were differences in the respiration between the sites that had not had fire history and the sites with fire history and fire severity. Where it was kind of nice. We observed differences between the top and bottom sections of the core for respiration, with an initial decrease at all sites between respiration in the top and bottom, as well as a decrease between the all sites between the initial burst respiration at 24 hours and at 96 hours in the top, and an increase in the bottom. I like to think of this is that the organic matter in the top section was highly metabolizable and is respired more quickly, where the bottom, it takes it a little bit longer to break it down. So it's less efficient for them to break it down. So it is consumed and respired after a longer time period. And then next, the organic matter was measured using FTI-CRMS and LCMS. And I'm going to be using the nominal oxidation state of carbon, which is an output of the organic matter composition, to as a numeric integrator or representation of sample chemical composition, as well as NOSC is related to oxidized and reduced members. The more negative a number is, the more reduced it is, and the more oxidized it is, the more positive it is. One of the major reasons why I'm going to be using NOSC for this presentation and to talk about the chemical composition and shifts is that fire is an oxidative process, and we work to look at fire as an incomplete or complete oxidization of carbon or organic compounds to CO2. And so with presence to fire, we would expect to see a nominal oxidation state shifting into the more oxidized or more positive. This figure is a little bit complicated, so we're going to work to set it up really quick. Focusing on the top sites, we have the lightest yellow in the solid line for the top sites followed by the orange and red representing no fire, moderate fire, and high severity burns. And one of the big things that we can see about this is that as we increase the burn severity in the top 10 centimeters, our nominal oxidation state continues to shift to be more oxidized or more positive. We observe a similar trend in the bottom 10 centimeters. However, we do not continue to see that progressive increase, and we see an increase in the nominal oxidation state more representative of burn history as opposed to burn frequency or severity. But again, as we have an increase in fire, we have an increase in the nominal oxidation state distribution. And then finally, I think one of the things that are really exciting is when we plot this data using a PCA, we get a separation into the unburned sites as well as the burned sites. And so that we can see that the PCA indicates that the soils with fire history are more similar than soils without fire history. As well as when we do a separation with depth, we observe that soils separate even with fire history into top and bottom depths. One of the reasons why I love this is that we were concerned that we wouldn't see depth distribution or depth differences on a 30 centimeter core, but we see this very clean separation in organic matter chemistry between the two depths that we're comparing. And finally, as we move into the X-ray computed tomography, what we can see is, so here's the X-ray scans of these different cores, and we can see that we have different densities and distributions of the organic matter, pores, and root structures. And when we move through this, we can compare the site with the highest fire severity. We can look at the initial image. We can then pull out what are the pores, just the connected pores, as well as we're working to develop and kind of finalize this pore network modeling. And what's really unique about this is we can look in WSU2, the one with one fire, and we can see that it has a large root structure, and we can actually model that preferential flow through that root structure that is part of the soil. And so it's kind of nice that we can capture some of these unique details or non-homogenized details. And so with this, we can see that we have in the unconnected or in the unburned site that we have many more pores and many more connected pores and higher flow through. And finally, when we start to look at these water retention curves, what we see is that there are differences in when the wilting point is and how these change. And what we find is generally higher clay content soils lose water slower and have higher volumetric water content at the field capacity and wilting points. We're working currently to scale these to meet the future MONet demands of these measurements take days to weeks. So this is kind of the slowest point for us. And then finally, as Emily mentioned, all of this is working to enter into a larger MONet database that will be open source and accessible upon completion of the database network. And then finally, I want to highlight the next steps for us are going to be to facilitate existing and future developer proposals within the national labs. Some examples of those are developing standardized workflows for XCT, as well as investigating some organic matter composition with TOF sims. We're preparing to release the data package for the collected pilot measurements. And as Emily mentioned, our goal for that is the end of the calendar year. As well as we have a user proposal call for MONet that John will present on, I believe. And finally, I wanted to thank kind of part of the bigger team that was part of this project. And then highlight that again, Emily Graham has led this project. And with that, I will default to John, who's next.
Okay, Roxanne, can you share it? Can you give me control of that screen again, please? Perfect. Thank you so much. And then go full screen. Okay, well, while I'm waiting for to get control of the screen, I'll go ahead and introduce myself. I'm John Bargar. I'm the science area lead for the Environmental Transformations and Interaction Group here at EMSL. And I'd like to follow up these very nice talks by Maggie and Emily and tell you a little bit more about the details of how you can get involved. I think this is the pointer options. I think that's laser pointer. Great. All right. So there's 4 major steps illustrated here. I'll go into detail in them more in just a minute. But basically, first, we need to open the sample solicitation call so you can send us your core requests. The second step is that you collect your samples and send them to us. The third step is that we analyze your samples. And then the fourth step is that we make your data available on the portal. I've added a 5th step to just really bring home the importance of the bigger perspective that Emily talked about a little bit in the first step, which is at the same time as you get a really amazing data that you can use for your own research projects for free. All you need to do is pay for the cost to go to the field and harvest the samples. You also are contributing to a much bigger effort, and you have the opportunity then to become really part of a much bigger project. Okay, so I'm just going to drill down a little bit to talk about these individual steps a little bit more. So in terms of opening the sample solicitation, we're hoping to do this in midwinter. After we do that, then you can fill out and submit a sample application. If you have formerly used the EMSL Nexus web portal for submitting proposals, you'll see that a lot of this will be very similar. We're going to be using the same basic process, but in this case, since we already know what measurements we're going to make, those are the ones that Maggie showed, it's like a fixed price menu. You won't have to check all those boxes about different measurements. You really are going to tell us about your sample site, and we need that information so we can do a technical review to make sure that the samples you're sending us are part of the regional study that we have open for the call at that point in time, and also to make sure that there aren't any administrative control boundaries that may create environmental compliance issues for us. So we want to make sure that they didn't come from a Superfund site and contain, for instance, mixed waste. And then the next step after we do the technical reviews, which we expect to be quick, we think these are going to be about 1 to 2 weeks, we will confirm acceptance through some kind of an email or notification from the web portal. So now the fun begins, right? You can collect a sample and send it to us. We do have a lot of activities that we need to schedule as part of this. When we receive your sample, we have to be absolutely sure that we have the capacity to accept it, dissect it, and immediately get the soils that you've given us into the -80 freezer for the DNA samples the same day, as well as initiate a couple of the other processes. So we need to schedule this carefully. And then after we schedule this, then we will send you the coring kit that Maggie just showed you. And then you send us a core, and that's really it. You'll need to basically pay for the travel to go and do this, but otherwise EMSL is covering the rest. So this really helps bring down the barriers to do this science. And that gets us to the third step, which is we analyze your samples, right? We dissect the core, as Maggie just showed. We perform analyses that Emily and Maggie talked about, and then your data are published on the Nexus MONet data portal. All right, so in terms of timeline, we're sitting here, right? It's almost October. And we're working really, really hard to try and get all the various working parts. There's a lot of pieces to this to make sure that we can we can begin to receive your sample requests in winter. Hopefully we'll have this open on February 1st. Please watch our MONet website for developing information about when we expect to open that. We're going to try to release the guidance for the call before AGU in December. So again, watch the website for evolving details. So let's say that it's the end of February and you submit a sample collection request. By the way, we're really trying to get this active so that we can begin to get cores collected in early spring so that we can get the entire next year season for core collecting. Then it's going to take, we expect, about a month between the time that you submit the application to when we can actually send you a core collection kit. So let's say that you do that and you collect a core and send it to us. Then as soon as the data are available and we've done the QC on it and we can get it onto the Nexus database, then the data becomes available to you to use. And we will continue to add more data as we get through the sample processing steps. Some of them, like metagenomes and the gravimetric water analysis, take longer because you have to wait for soils to dry. So some of these processes are inherently slow. The CT, we have to measure the whole core, that's limited. But nevertheless, we really expect that within 6 months, we should have all the data to you, if not sooner than that. That's one thing that we'll be working on. And then of course, the data will become, eventually all your data will be available on the web portal. Just waiting for the slide to advance here. I'm a little bit afraid to hit advance one more time because then it's going to overshoot. All right, so those are kind of the timeline and the breakdown of the major steps. I also want to tell you guys a little bit more about accessibility, who can participate, and some of the underlying guidelines that we're adhering to. So the first point I'd like to make is that the MONet program is truly a user-enabled science. Oh, I'm trying to get my laser pointer back, but I guess I'm going to skip it for now. It's very, very tiny on the screen. So it's open to individuals at universities, so graduate students and postdocs and faculty members. It's available to scientists in national laboratories. It's also accessible to individuals who are citizen scientists. We are also encouraging large program collaborations. We are going to suggest as much as we can that if it's possible for you as an individual to collect samples at existing intensive sites where there is a good infrastructure footprint and there may be historical data, for instance, with greenhouse gases, and there's likely to be continued commitment to sampling in the future, we will encourage folks to consider reaching out to those sites and using them. But we're also really interested in the geographic diversity that comes from individuals collecting samples near their home institution. And the other thing that's really important to emphasize is that we're building this database to really minimize barriers to participation and barriers to access to the data. So that means we're following so-called FAIR principles for the database. FAIR stands for findable, accessible, interoperable, and reusable. And one of these practices means that your data will become available to the whole community at the same time it becomes available to you. Now, some people may think, oh gosh, I'm really worried about participating because I'm afraid that somebody might scoop me. And my response to that is, please don't worry about it, because in order for that individual core or set of cores from that individual site to be useful to you, you need to have an enormous amount of domain knowledge, as well as hypotheses developed for that site, which other people just aren't going to have access to. All the specific site conditions that are extant that allow you to formulate a specific hypothesis or hypotheses and test them. I mean, that's in general, other people aren't going to know that. They're not going to be able to get in and grab your data and scoop you. However, what will be possible is modelers who want to take these data from individual sites and aggregate them up to the regional scale will have access to those data so that they can continue to improve the amount of data and decrease the uncertainty of the model. So this is why it's very, very powerful and important to have this type of access. There's 4 major types of sample solicitation programs that we're currently planning to release. The first one is regional soil function, where we're going to be looking at the continental United States and looking at the neon ecological domains and then binning them into subdomains. And we'll probably be requesting at any one point in time for cores to be submitted to us from 2 or 3 of these domains. And we're kind of go region by region over the next following years to build up the map. The other thing that we're going to do is have a soil perturbation rapid response call so that we can move quickly to seize scientific opportunities that are created by large flood events or burn events or other types of events where there's a lot of kinetics and very quick responses that are happening within a few days after the event. And of course, then there's a sustained response. So this should allow us to address those as well. We also are interested in setting up long-term regional study sites and also user-driven focused research campaigns, which will be focused on testing specific hypotheses that are developed through community discussions. These 2, because they require quite a bit more socialization, we won't be able to roll these out as quickly as the first 2, but our goal is to indeed roll these out within the new fiscal year. So that would be fiscal year 23. So I would just like to, go ahead, Roxanne, and roll this video. Everyone in the audience, could you please turn up your volume on your speakers so that you can hear the sound? This is a really cool video from the pilot project that Emily led. And you'll see Emily and Maggie and their field team out at the Prosser field site collecting samples. It's just 30 seconds, so it's really quick. Fantastic. And if you're wondering, yes, Emily was about 7 months pregnant at the time. And no, it was not necessary to fill out 2 permits in order to get access to the field site. So that concludes all the slides that we wanted to share with you today. And at this point, we can stop and I'll turn it back over to Linda to take questions. And Linda, do you want us to all come back online with the video now?
Yes, all 3 of you. And Roxana will spotlight the 3 of you across the screen. So since we're limited on time now, we got about 12 minutes for questions. So, and we've got quite a few questions. Let's try and keep them as the answers as brief as possible. So I'm going to start, unless I say who it's for, just somebody take the question. So the first question is from Pitar Penev. What material is the yellow fixture in the cores from slide 14 made of? And I'm guessing that's you, Maggie.
Yeah, it's a polyethylene foam. It's a solid core pool noodle.
Cool. Next one's Taylor Siley. Hey, are the standardized analytical methods used for Thousand Soils project listed online anywhere or published? Curious if the approaches we're currently using in our lab match up.
Yeah, we are in the progress of getting those published on protocols.io. And so they should be published and publicly available by the end of the year.
Awesome. So Asif Redon, great talk. Can this pore structure analysis be applied for materials like activated carbon and biochar?
Yes.
Yeah, I might defer to John. We do have a micro CT that's not what we standardly use if you want higher resolution. I think right now we go down around 30 to 50 micron resolution. So it depends on whether or not you want to look at, small chunks of biochar or a full core.
Yeah. The short answer is yes. Yeah.
All right, cool. So the next question is from an anonymous attendee. How is the variability within site condition, and how many samples per area do you recommend we sample?
What I will say is the variability is higher at surface soils, and the number of samples that you should be collecting is a bit dependent on your research question. If you're looking at continental scale, you can have fewer replications within one site, but if you're looking at a single site or a specific site, you should have higher replication. And there's no real perfect number for that.
All right, next one from Natalie DeHerby. Hi, thanks for all the amazing work. When and where will the results of the pilot be accessible? Thank you.
So by the end of the calendar year is our target. And so we have this webinar registrant list. We will email that out. We have someone working on it at the moment. We're looking either at our internal database called Data Hub or ESS Dive as a final package delivery. And we're finishing up the analyses from the last set of cores right now. So I think end of calendar year, early next year is a good target for that.
What I will say, the only data that won't be completed by then is the hydraulic properties.
Okay. Next question, for the PCA analysis of organic matter chemistry with respect to different depths, do you see differences in unburned samples?
When we look across burn samples and unburned samples, we see those differences, but this subset of samples does not have enough to cluster them by burned top and bottom and unburned in a four-way kind of analysis for that. But we do have more samples with fire, and so our goal is to include that kind of bigger picture elements of those groups once we have more sample numbers.
All right. What method is used for the chemical analysis of macro and micronutrients?
So there's quite a few, and in the flyer, there's a little bit more detailed method information. So that should be coming out next week. We measure the macro and micro elements through a combination of ICP-MS, carbon and nitrogen, as well as phosphorus and nitrogen extracts that are measured either through colorimetric or what's the other one? Other chemical analyses. I can't remember what the exact instrumentation for it is.
Yeah, all of those will be in detail on the flyer that we'll send out. We also use like an elementar for total carbon and total nitrogen. They're all standard methods that the community tends to use.
All right. I think this was already partially answered, but could the speakers remind us at the end who the PIs are, which are developing the field sensors and the rhizosphere sensors for the full MONet network?
John, do you want to take that one?
Sure, it's Amir Akami. And we'll put that name in the chat. I'll do it just a minute.
Okay. So the next one, are you planning to fill up the database with data processed only through EMSL or contributions from different labs?
So only through EMSL, or we have some contractors that do that, like the standard chemical analysis for us. So it'll be either through EMSL or EMSL contracted vendors, and everything is standardized across all those. So every sample is treated the same way. So they should be directly comparable that way.
Great. Hi, I have two questions for Maggie. how long did it take to generate XCT scans for the soils under different burn severity?
Yeah, so the scans are actually very quick. You can do multiple in a day. They take an hour or two, depending on how kind of complicated the soils are, but the soil processing and the image processing and slicing and recombining for XCT is the time consuming part. And so that, depending on how complicated it is to stitch it back together, it can take hours to days to weeks, depending on how many replicates or how many soils we have, or just kind of the complicatedness of the profile. But we are working on some methods to kind of help our XCT team speed that up.
Great. Are the CT scanned sections usable for other analyses?
Yeah, so as part of the database, our plan is to provide the raw images that can be read in, and they're complex. And if you can work with them in that format, that's great, as well as some refined and standardized data products.
Okay. Wondering how many cores are allowed from a given collaborator and or given spatial domain. Also, can additional data types be requested beyond EMSL's consistent set?
Yeah, I would, if you have a number in mind, I would suggest you work with us on that. We don't have a hard limit, but we do want to have a bunch of different collaborators participate. And some of these, as Maggie noted, are less high throughput than others. So on the exact number, just work with John or I on that, or Maggie on that. And yes, in a limited fashion, you can suggest other measurements that might help you address more specific hypotheses.
Great. Also curious what values were fed into the PCA, the NOSC values, other calculated soil properties.
So for those PCA values, we used all of the assigned peak values. So not the derived NOS products, but the essentially presence absence peak data. So those separations are taking into account all of the peak data.
Okay. Would agricultural soils be considered by MONet, or is it focused on natural ecosystems?
We are allowing agricultural, urban, disturbed. We don't currently have a focus on just natural ecosystem soils. We actually have quite a few agricultural or treatment soils.
Okay. Are you accepting samples from outside the US, like Canada?
Right now, we're focusing on samples from within the US and Alaska because we're starting up. We have limited resources for now. Our intent is to expand beyond the borders of the US when we can. But for now, we just are focusing on US.
Can we propose to explicitly work with specific modelers in our sample solicitation?
Absolutely. I'm sorry, Emily, do you want to answer this? I didn't even think.
Yeah, no, that was my initial response as well. Absolutely. And if you don't know who to work with, Satish Kara, who's our computational IRP lead, is a great person to work with to point you to the right person on that.
Okay.
Can I just add to that?
We are getting down to the wire, but there's still quite a few people here, so that are still tuning in. A few more questions. I'm interested in permitting aspect of taking cores and who has the responsibility for securing those permits. Is EMSL helping folks to obtain the right permissions?
Do you want me to answer that, Emily?
So this is run through our user program. So the permitting process is the same as through our user program, where we expect that individual researchers have the appropriate permit to collect the cores.
Great. I'm looking at the EMSL website, and there's a survey for 1,000 Soils Research Campaign at EMSL on the MONet page. Is the survey for submitting soils from our site?
Yeah, so the survey that is on there is a participation inquiry. Any of those that get filled out are going to be redirected to the MONet proposal launch that we'll be having. So that we should probably remove that survey.
Oh, okay. So will this be available for viewing again? One of the students in my lab is not here today and he should see this, and I'm going to answer this question. And yes, a copy or this recording will be available on EMSL's website under the EMSL LEARN section. And so I believe that Genoa has been dropping links, but if you can drop it one more time in the chat, Genoa, the link to the EMSL LEARN site, you will be able to find it there in about a week. So the next question, can you explain a bit more about rhizosphere sensors? Briefly.
You wanna-
John, I think that's you.
Okay, no, you can do it. So rhizosphere sensors. So there's many answers we could give. So first of all, the idea is to detect. I mean, currently there are sensors that can be deployed in the rhizosphere that can be used to detect all kinds of things like soil moisture and even redox. And there's slightly more chemical sensitivity than that that's coming online. So we are not interested in competing with off-the-shelf commercial vendors or people that are developing products that will be commercialized through off-the-shelf commercial vendors such as Fondriest or Campbell Scientific. Rather, what we're looking at is, can we detect specific organic molecules, small organic molecules that are present in the rhizosphere that are commonly used for signaling between, say, plants and microbial cells, that can be leading indicators, or they can be leading indicators of change, and they can also be indicators of response to environmental perturbations. And so it's a significant R&D effort. It is a decadal vision. So what I'll say is we are early on. Having said that, we actually have or are taking several active proposals right now in our internal S&T research portfolio that are beginning to propose methods that will do this, for instance, by deploying a spatially resolved high resolution mass spec. So you can go in and take very tiny particles and you can get a very high resolution measurement of the chemical formula of that material. So
All right. We've got a couple more questions, but I think we will email the folks who have these questions individually. Most of them come from James Stegan, who we know quite well, so we can answer him directly. And there's a couple others, but Eric, we will get back to you on your question as well. So we're over time, and thank you everyone for joining us today. We're excited to work with you on this incredible initiative that we've started, and we hope you have a fabulous rest of your day. Thank you.